Okay, my dear friends, this is Dr. Padala Thirupathi, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal. మనం ప్రీవియస్ పేపర్స్కి సంబంధించి ముఖ్యంగా మనకు టీఎస్ సెట్ ఇంతకుముందు 2012, 13 అదేవిధంగా 14లో జరిగినటువంటి, అదేవిధంగా 16లో జరిగినటువంటి, 19లో జరిగినటువంటి ఈ సెట్ ఎగ్జామినేషన్కి సంబంధించినటువంటి ప్రీవియస్ పేపర్స్ విశ్లేషణ చేసుకుంటున్నాం. దాంట్లో భాగంగా ఈరోజు మనం టిఎస్ సెట్ అంటే ముందు ఏపీ ఉమ్మడి ఆంధ్రప్రదేశ్ ఉన్నటువంటి క్రమంలో మనకు నిర్వహించబడినటువంటి ఏపీ సెట్ కు సంబంధించినటువంటి ఏపీ సెట్ 2012 కు సంబంధించినటువంటి పేపర్ 3ని మనం విశ్లేషించుకుంటున్నాం. అయితే ఇక్కడ ఒక విషయాన్ని మనం గమనించాలి. పేపర్ 3, పేపర్ 2, పేపర్ 1 అనేటువంటిది గతంలో ఉండేది. ఇప్పుడు టిఎస్ సెట్ మనకి ఆన్లైన్లో కానీ ఆఫ్లైన్లో కానీ ఉండవచ్చు, అంటే కంప్యూటర్ బేస్డ్ టెస్ట్ ఉండవచ్చు. అయితే ఇందులో పేపర్ 1 టీచింగ్ ఆప్టిట్యూడ్, రీసెర్చ్ ఆప్టిట్యూడ్, అదేవిధంగా మెంటల్ ఎబిలిటీకి సంబంధించి ఉంటుంది. పేపర్ 2 అనేటువంటిది కన్సర్న్ లైఫ్ సైన్స్ లో ఉంటుంది. పేపర్ 3 ఉండదు, కానీ ఇది గతంలో నిర్వహించినప్పుడు పేపర్ 3 ఉండేది. ఈ విషయం మర్చిపోవద్దు. అభ్యర్థులు ఇది గుర్తుంచుకోవాల్సినటువంటి ముఖ్యమైనటువంటి అంశంగా మీరు భావించాలి. ఓకేనా? రైట్. మరి ఇది 2012లో జరిగినటువంటి లైఫ్ సైన్స్ కి సంబంధించినటువంటి 2012లో జరిగినటువంటి లైఫ్ సైన్స్ సంబంధించినటువంటి ప్రీవియస్ పేపర్ మనం విశ్లేషించుకుంటున్నాం లైఫ్ సైన్సెస్ కి సంబంధించి. సో దిస్ ఇస్ ద పేపర్ 3 ఇన్ ఎర్లియర్, బట్ నౌ అంటే ప్రెసెంట్లీ మనకు పేపర్ 2 మాత్రం ఉంటుంది. పేపర్ 1 టీచింగ్ ఆప్టిట్యూడ్, రీసెర్చ్ ఆప్టిట్యూడ్, పేపర్ 3 ఉండదు. పేపర్ 2 ఈ లైఫ్ సైన్సెస్ సంబంధించి ఉంటుంది. అంటే బాటనీ, జువాలజీ, బయోకెమిస్ట్రీ, బయోటెక్నాలజీ, మైక్రోబయాలజీ, ఎన్విరాన్మెంటల్ సైన్స్, ఈ స్ట్రీమ్ లో ఉన్నటువంటి వాళ్ళు ఈ ఏపీ మన టిఎస్ సెట్ కి సంబంధించి లైఫ్ సైన్సెస్ ఎగ్జామ్ రాయడానికి అర్హులు. ఈ విషయం మర్చిపోకూడదు. రైట్. ది ఫస్ట్ క్వశ్చన్, ఇందులో మొత్తం మనకి ఈ ప్యాటర్న్ ప్రకారం ఇందులో 75 క్వశ్చన్స్ ఉంటాయండి. దీనికి సంబంధించి మనం చూద్దాం. ద హైయెస్ట్ కాన్సన్ట్రేషన్ ఆఫ్ సిస్టీన్ కెన్ బి ఫౌండ్ ఇన్. సో ది హైయెస్ట్ కాన్సన్ట్రేషన్ ఆఫ్ సిస్టీన్ కెన్ బి ఫౌండ్ ఇన్ కెరాటిన్. సో కెరాటిన్ ఇస్ a protein. Keratin is a protein. Cysteine is an amino acid. So this keratin protein contain highest concentration of cysteine, that is amino acid. The second one is the membrane bound enzyme that catalyzes the formation of CAMP from ATP that is found adenyl cyclase. Okay. Then this is assertion and reason. So these type of questions definitely we are observed in the all set, even though the CSIR net examination also. So the first assertion is assertion or assertion. This quorum sensing is the ability of bacteria to communicate and coordinate behavior. This is assertion. And what is the reason? The reason, bacteria those use quorum sensing to produce and secrete a signaling chemical called inducer. So this question about this assertion and reason, both A and R. So assertion and reason both are true, are correct, and R is correct explanation to A, that is assertion. Then fourth question, this is also same like third question. Interferons are the special type of proteins. Remember here, interferons are produced by the host against viral infection. Okay, so during viral infection, the body of the host cells, maybe the human beings or maybe the plants or maybe the animals, they are produced antiviral proteins or antiviral special type of compounds that we can call as interferons. Okay, so this is A is absolutely correct, but R is interferons are essential for viral replication. So this is not true, my dear friends. Only assertion is true. Only A is correct. And the fifth question, cyclomicrons, intermediate density lipoproteins, that is ideal, intermediate density lipoproteins, ideal, and LDL means low density lipoproteins, and very low density lipoproteins, that is VLDL, very low density lipoproteins. All are serum lipoproteins. what is correct ordering of these particles from the lowest to the greatest density? So according to the density, which one is from the lower and which one is the higher? So compared to, see here, my dear friends, cyclomicrons, from cyclomicrons, see here. So first cyclomicrons, then VLDL, very low density lipoproteins. Okay, very low density lipoproteins. Then only ideal, intermediate. See here, intermediate density lipoproteins, that is ideal, intermediate density lipoproteins, then LDL, low density lipoproteins. So these are the correct sequence of the based on the density, lowest density to highest density, Okay. So the next question, the sequence of the steps involved in the viral protein, viral replication. So viral replication, the correct sequence of these steps involved in the viral replication. So viruses are obligate parasites. Viruses always, they are infect other living organisms like human beings, animals and plants. So viral plant viruses, animal viruses, and even the human viruses. And remember, my dear friends, any virus contain only one type of genome, that is RNA, either RNA or RNA. Only one type of genome or one type of nucleic acid is present, whether DNA or RNA. Okay. And remember the sequence of the viral replication or life cycle of the viral replication or life cycle of the lytic life cycle. Especially we are going to discuss about the lytic life cycle. See here, this is the sequence. Whenever some of the bacteriophages, they are attack on the Escherichia coli or other bacteria member, the bacteriophages or viruses, they are attack on the bacteria. So in this bacteria, first step is attachment. Attachment means absorption of the surfaces. ఈ విషయం మర్చిపోకూడదు. ఓకేనా? Absorption of the surfaces. Okay. So whenever the bacteriophage is going to attach to the surface of the bacteria, then the step we can call as adsorption. That is nothing but attachment. The second step is penetration. Penetration means the bacterial genome or bacterial DNA inserted into bacteria. That stage we can call as penetration. And the third one is uncoating. That means the bacterial genome is going to enter into the bacterial cell. Then automatically it controls the entire bacterial mechanism or bacterial metabolic activities, and it will replicate to produce the new naked DNA particles or naked DNA segments or fragments. That step we can call as uncoating. And then whenever the uncoating process, the genome replication and expression. This is the last step. Replication and expression, which means it multiplication. It produces number of DNA replicas of the bacteriophage. So these are the steps. First step is attachment. The second step is penetration. Third step is genome replication and expression. And third one is the uncoating. The fourth one is genome replication and expression. And the seventh one is in the area of desmosome, the adjacent cells have desmosome. See here, my dear friends, desmosomes, the adjacent cells have. Number one, remember, here intercellular thickening material and also the transmembrane linkers. Say both they have desmosomes. Okay, so desmosomes very nearer to the adjacent cells which have intercellular thickening material and also transmembrane and linkers. Okay, this is seventh question. And the eighth one, viruses contain the following. Remember, my dear friends, the virus contain genome, then only capsid, and along with enzyme is present. So these are virus particles contain this genome, capsid, and enzyme. Capsule is present only bacteria, not viruses. Okay, capsule not related to virus. And the ninth one is DNA polymerase 1. DNA polymerase 1 are multifunctional enzymes because, remember, my dear friends, during DNA replication or even though the transcription, so these polymerases, especially DNA-dependent RNA polymerase, transcriptase is nothing but DNA-dependent RNA polymerase 3, RNA polymerase 2. So like that, DNA polymerase are the most important enzymes. Okay, see here, these DNA polymerase 1 are multifunctional enzymes. Why? Because number 1, polymerization reaction. Number 2, removal of the nucleotides from 3' and Q terminus in DNA, and removal of nucleotides from 5' terminus in DNA. Okay, so these are multi-functional enzymes. That's why DNA polymerase 1 is called as multifunctional enzyme. The 10th one, most of DNA binding proteins insert the following structure into the major groove of DNA during binding. So that is alpha helix. Then 11th one, coding region of an mRNA is 336 nucleotides long, including the initiator and termination codons. What is the number of amino acids in the protein translated from this mRNA? The correct answer is 111. The correct answer is 111. And the next one, origin of replication, that is ORI region. Origin of replication means ORI region, origin of replication site. So ORI region or origin of replication E. coli consists of, number 1, repetitive elements along with 80 rich regions. 80 means adenine and thymine. 80 rich regions, ORI regions possess rich regions of the adenine and thymine regions, that is 80 regions, and repetitive elements. And Bt cotton contains, remember my dear friends, Bt cotton contains Bt genes or Cry genes, Cry1AC, Cry2AB, Cry1AC and Cry2AB. These two types of the crystalline proteins or crystal genes or Cry genes are present in Bt, that is Bt cotton. This Bt cotton is especially lepidopteran, like bollworms, these type of insects resistance genes. These genes are kill the insects, especially larva stages. Okay. Then choose the correct signal transduction pathway. Signal transduction pathway. What is the correct option? See here, hormone, 7-TM receptor, G protein, CAMP, adenosine monophosphate, and pKa. So this is the correct pathway for the signal transduction. And the 15th one, which of the following can form a part of biosensor? That is enzyme and antibody. Okay, enzyme and antibody can form the part of biosensor. Okay, biosensor or SPA. Biosensors are formed in the part of biosensor formed with the help of enzyme and antibody. Then 16th one, name the enzyme act as intracellular mediator for growth factor receptors. That is JAK kinase or JAK kinase or JAK kinase is the right answer. Then 17th, DNA can be radio labeled with DNA. Remember, my dear friends, DNA radio labeled with definitely P32 and P33 radioisotope. And yes, 35, especially we are observed in especially in DNA, which means especially the nitrogen and phosphorus. Okay, so phosphorus labeled with DNA phosphate. DNA phosphate is the skeletal for the DNA structure. So the DNA is identified with labeled P32 and P33 labeled isotope. Okay, we can use. And the 18th one, electrophoresis of the purified protein called X in the presence of SDS can, and beta mercaptoethanol shows a signal band of 60 kilodalton In gel filtration experimentation protein, X elutes between alcohol dehydrogenase, that is 160 kilodalton, and beta amylase 190 kilodalton. How many identical subunits in the protein? X composed. That is 3. That is 3. Then branched hydrophobic amino acids are. Branched hydrophobic amino acids are. Number 1, valine, leucine, isoleucine. So among these 4, first 3 amino acids are branched hydrophobic amino acids. Valine, leucine, isoleucine. These 3 are the branched hydrophobic amino acids. Okay. Then next one, that is 20th question, assertion and region, A and R. Olfaction and taste are important sensory modalities for insects, particularly in mating, oviposition, and food selection. Then R is chemoreception in insects is well established than the other animals. So both are correct, my dear friends. At the same time, R is correct explanation to A. Okay. Then this is the match for the jaw suspension type of various organisms. Jaw suspensions in various organisms. That is autodiastylic. Autodiastylic, that is bony fishes. Amphistylic, that is a shark. Hyostylic, elasmobranches, elasmobranch fishes. And autostylic, that is mammals. Okay, that is jaw suspension types. And 22, arrange the transport pigments in the order of the evolutionary origin from more recent. That is hemoglobin, more recent, from evolutionary origin from more recent. Hemoglobin, myoglobin, chlorocruokin, and hemocyanin. Hemocyanin. So this is the correct order from the evolutionary. Evolutionary from lower to higher. So from recent. Okay. So this is the 22nd question. And 23rd question, that is match the following. Then the 23rd question, that is match the following. List 1, that is genes, and list 2, that is representative characters. So which gene is responsible for which character? So that is incomplete dominance. Pink color of the 4 o'clock plant. Pink color of the 4 o'clock plant. So Mendel, Gregor John Mendel, has unable to explain this incomplete dominance, my dear friends. Remember. And codominance. The codominance, human being belonging to AB blood group, that is codominance. Incomplete dominance, 4 o'clock plant, pink flower color. And the codominance related to human being belonging to AB blood group. Actually, AB blood group of the human being. And the third one, polygenes. So polygenes is responsible for the human color of the human beings. So color of the human beings responsible for the polygenes. And the complementary genes. This is the last option in this 23rd question. The complementary genes are responsible for the purple color in maize due to anthocyanin. We are observed the multicolors or different colors in maize or corn plants. So that corn kernel shows different colors of the maize seeds just because of the complementary genes. Okay, so this is the most important question related to this set examination and also other competitive examinations, my dear friends. And the 24th question, the crossover percentage between linked genes, that is J and M, is 20% J and L is 35, and J and N is 70%, L and K is 15%, M and N 50%. Thus, the sequence of genes on the chromosome is. So the sequence of the chromosome, that is J, M, L, N, K. Okay. Then 25th question. Mammals and birds were originated in which of the following era or epoch or period? So generally mammals, though highly evolved organisms in the chordates, especially mammals and birds, these are evolved from the Jurassic period. Okay. Evolved or originated from Jurassic era or Jurassic period. Then the Hardy-Weinberg law. This is the most important law regarding of the community ecology or the especially in the population ecology. The Hardy-Weinberg law is expressed as P plus Q square is equal to RQ, where P and Q stands for. It is very equal to the A plus B whole square in mathematics. Like that, P plus Q whole square is equal to P square plus Q square plus 2AB. So this is the equation of the P plus Q whole square. Then P and Q stands for. P and Q stands for gene frequencies in population. Gene frequencies in population. And 27, that is A, and means assertion. Autonomous specification gives rise to pattern of embryogenesis referred to as mosaic development. If particular blastomere is removed early in its development, that isolated blastomere will produce the same type of cells that it would have made if it were still part of the embryo. The embryo appears to be constructed like a file mosaic of independent self-differentiating parts. So this is R. Both A and R are true, and R is correct explanation to the A. R is correct explanation to R. R is correct explanation to A. Okay, so this is about this question. Okay. Then the 28th question, that is assertion and reason. Assertion: structures of globular proteins are accurately determined with X-ray diffraction, and R, wavelength of X-rays is less than 2° angstrom unit, and the resolution of structural information achievable is around 2 angstrom unit. So according to this question, A and R is correct. Both are correct. At the same time, R is correct explanation to A. Okay. Structure of globular proteins are accurately determined with X-ray diffraction. Wavelength of X-rays is less than 2 angstrom unit, and the resolution of structural information achievable is around 2 angstrom units. And the 29th question, that is A and R, assertion and reason. Among these two statements, we are observed. Among these two statements, A, foreign genes can be cloned into plasmid. Foreign genes cloned. Foreign gene cloned can be cloned into plasmid cloning vectors, and cloning vectors can be used for expressing the foreign genes in bacterial system. Among these two statements, A is correct, but R is not correct. A is correct. That is, foreign genes can be cloned into plasmid cloning vectors. Plasmid cloning vectors. Okay. And the next question, 30th, that is assertion: plant breeders have an advantage over the animal breeders, and reason, that is, plant breeders can employ clonal propagation. Both are correct, and R is correct explanation to the A. Okay. Plant breeders have an advantage over the animal breeders. Plant breeders can employ clonal propagation. So both are correct, and R is correct explanation to A. And identify the correct statement. Wendt's theory explains the reasons for geotrophism. Okay, now Chladni-Wendt theories. Chladni-Wendt theory. Actually, Chladni-Wendt theory is clearly explained that what is the reason? The reason is of the geotrophism. How roots grow towards the soil or inside the soil. That the entire physiological activity explained by Chladni-Wendt theory. Okay. Then 32 question. Then 32 question. In these two statements, several factors are responsible for seed dormancy. One of the factor is immature embryo. Yes, absolutely. A is correct. And seed dormancy caused by immature embryo can be overcome by scarification. Can be overcome from scarification. No, it is wrong answer. This is overcome by actually overcome by embryo culture in plant tissue culture. Okay, but the scarification is very related to seed dormancy overcome by the hard seed coat, especially in Fabaceae members like pulses, all pulses, soya, bean, and other beans. Okay, that is scarification, very related to pulses family or Fabaceae family members. Then the 31 question in this among these two statements, these are the terms related to definitions. Photooxidation. Photooxidation means the damage of the cells under the high intensity of the lights. We can call it as photooxidation. Okay, photooxidation means high intensity of the light. The cells are going to damage. And photoperiodism. Photoperiodism is coined by or given by Garner and Allard. Garner and Allard has given the photoperiodism. The photoperiodism defines that influence of the duration of day and night on flowering plants. So actually the critical day length is 12 hours for the during day hours and 12 hours for the night. But based on this photoperiodism, plants are divided into long day plants, LDP, short day plants, SDP, and DNP, day neutral plants. So this photoperiodism is given by Garner and Allard scientists. Both the scientists have given this photoperiodism. And photolysis. This photolysis involved or absorbed. Photolysis absorbed in splitting of the water molecule by light. Okay. Photolysis of water absorbed in the photosynthesis during the photosynthesis process, splitting of water molecules by light. Then photorespiration. So this photorespiration, we are absorbed in the peroxisomes. Absorbed in peroxisomes, respiration in chloroplast during daytime. That we can call as photorespiration. So this photorespiration takes place in three cell organelles. That is chloroplast, mitochondria, and peroxisomes. These three cell organelles are responsible for photorespiration. Okay. So especially the respiration in chloroplast during daytime. And 34th, the light-sensitive plant pigment phytochrome was first isolated by. Very important question, my dear friends. So the phytochrome, it is a pigment which is very light-sensitive pigment. So phytochrome is a light-sensitive pigment which is first isolated by Butler and his co-workers or colleagues. Butler et al. Butler and et al. These are the scientists and co-workers. They are isolated firstly of the phytochrome pigments from the plants, leaves, which is phytochrome is very sensitive to the lights. And the 35th question, which one is the not correct statement? Remember, my dear friends, whenever we are going to opt to the answers, should be clearly study about identify the question, whether it is not correct statement or correct statement. So in this question, which one is in the following not correct statement? Among four statements, which is not correct statement? So calcium is a component of chlorophyll. My dear friends, remember, calcium chlorophyll does not present in chlorophyll. Magnesium is present in chlorophyll, my dear friends. Magnesium, Mg+2. And this magnesium ion is responsible for the making of this entire chlorophyll porphyrin or porphyrin structure. And magnesium is ion responsible for the combining of the two subunits of the ribosome. And iron is a structural component of porphyrin molecule. Yes, we know that iron is a structural component in porphyrin molecule, in especially hemoglobin. And boron plays important role in translocation of sugars. In phloem, the sugar translocation done by boron pigments, boron micronutrients. And molybdenum. Molybdenum plays an active role or vital role in the nitrogen fixation. Okay. And the list of some terms and list of some two, these are definitions. Nitrification. So this question related to nitrogen cycle. My dear friends, remember those who are preparing NET, SET, or JL, DL, and even though PG entrance examination, this question is mandatory. Without this question, there is no set, AP set or TS set, or even though CSR, UGC CSR net, even though CP get examination or CU set, common entrance examination of the central universities, any examination, this question should be there. Remember, my dear friends. Nitrification, that is conversion of the ammonia into nitrates, not nitrates, nitrates. And denitrification means conversion of nitrates to gaseous nitrogen or atmospheric nitrogen. The nitrogen fixation is the conversion of the proteins to ammonia. Conversion of proteins to ammonia. That is nitrogen fixation. Sorry, my dear friends. Nitrogen fixation is conversion of gaseous nitrogen into usable form of the nitrogen. That is ammonia. Plant usable form of the nitrogen, that is ammonia. Then ammonification. Ammonification means conversion of proteins into ammonia. From ammonia, proteins are formed during ammonification process. Then the 37th question, the following proteins are involved in protein synthesis in ribosome. Protein synthesis in ribosome. Okay, that is a 37th question. The correct answer is here. Among these protein synthesis, these are the factors involved. These are the proteins are involved protein synthesis. EFTU, elongation factor. RF1, that is RF1, RRF, and EFG and IF, initiation factor. And the last one is RF1, RRF. The correct statement is 51423. 51423. See here. 51432. So these are the sequences. 51423 is the correct sequences. Okay. So the protein synthesis during photosynthesis, IF2 is the first involving during the photosynthesis, and the second one is EFTU, and third one is EFG, and fourth one is RRF, and last one is RF1. So these are the correct statements. Okay, correct sequence. Involve translation process in cytoplasm. Transcription takes place in nucleus. Translation takes place in cytoplasm. These are the correct sequence. That is IF2, then EFTU, then EFTU, okay, then EFG, so that is EFTU. The second one is EFTU, and third one is EFG, and then fourth one is RRF, and the last one is RF1. So this is the correct sequence of the 37th question. And then next one, choose the correct sequence of the following tools applied proteomic. So in this proteomic process, the correct sequence is isolation of the proteins, then IEF, and third one is SDS page, and last one is MALDI-TOF, MALDI-TOF. Okay. And then next one, then 39th question. 5 proteins with the following pi value were separated by pi value, that is pi value were separated isoelectric focusing, that is, which of the order of the migration from the anode closest to farthest. In this one, so second 2.1, see here my results. 2.9, then 2.9, then 3.6, then 6.5, then 7.2, 7.2, last one is 8.5. So this is the correct order. Okay. Two 2.9, 3.6, 6.5, 7.2, and last one is 8.5. So this is the correct sequence, correct order for the migration from the anode. Okay. The next one, the 40th question. Somaclonal variation. We are reached or we are produced somaclonal variations from callus culture and haploid culture using anther culture and protoplast isolation, that is cell wall degrading enzymes, and secondary metabolite production, suspension culture. So suspension culture used for secondary metabolites production and the cell wall degradation enzyme by protoplast isolation. Okay. Whenever protoplast isolation, we should degrade the cell walls. Okay. Then the haploid plants are produced through anther culture and somaclonal variations are produced through callus culture. Okay. This is related to plant tissue culture. And the 41 question, column A and column B. So among these, plasma membrane is 5 nucleotidase and nucleus DNA polymerase, mitochondria SDH, and microbodies catalase and peroxidase. These are enzymes which are present in that specific areas. So mitochondria consist SDH. Okay, SDH. And microbodies, they are possess catalase and peroxidase enzymes. Nucleus contain DNA polymerase enzyme, and plasma membrane, that is consists 5' nucleotidases. In the 42 question, which of the following pairs is not matched correctly? So the right answer is type of molecular marker YAC. Okay. YAC. Which of the following pair is not matched correctly? So type of molecular marker YAC is not correctly matched. Remaining are correctly matched. See here, production of recombinant protein, that is expression protein, vector. Immunological detection method, that is ELISA, enzyme-linked immunosorbent assay. Enzyme-linked immunosorbent assay, that is immunological detection method. And measurements of radioactivity by scintillation. Scintillation counting is used for the measurement of the radioactivity. And allosteric enzymes are ATKH, isoenzyme, that is LDH, the multi-enzyme complex, that is PDC, and coenzyme, that is acyl coenzyme A. And the 44 question, which of the following pairs is matched correctly? That is protein involved in elongation of translation, EFTU. This is correctly matched. Okay. Protein involved in elongation of translation, that is elongation factor, translation unit. And the 45 question, which of the following pairs are not pairly correct? So this is not correctly pairs. Remaining 3 are correctly paired. So the correct answer of this question is A, but correctly paired here, transposons identified in Drosophila, that is elements, tandemly repeated elements, that is VNTRs, and mobile elements found in base, that is ACDS elements. That is also we can call as transposons. Then assertion and reason, amino cyclation of tRNA is a very accurate process. That is true. That is true. Okay. Amylation of tRNA is a very accurate process. Definitely. Then R is wrong. What is R? Proofreading does not occur during amino acylation of tRNA by amyloacyl tRNA synthesis. So R is false. Then 47 question. Lac operon is activated in bacteria by the presence of lactose in the medium. Yes, definitely without presence of the lactose, lac operon does not activate it. So lac operon activated whenever there is presence of lactose in the medium. But R is false. That is, lac operon is not affected by the glucose concentration in the medium. This is wrong answer. Okay. Then 48 question. A monolin is a sweetener and it is a protein. When it is heated, its sweetness is lost. Okay. So it is right answer. A monolin is a sweetener and it is a protein. When it is heated, the sweetness is going to last. Then R, the reason for its sweetness lies in its conformation. So both A and R are correct. Then 49 question. In a flower, odd sepal is anterior. Petals arranged in a descendingly imbricate estivation. This flower belongs to Fabaceae. That is Leguminaceae family members. Fabaceae. Okay. So in this Fabaceae, especially the papilionaceous corolla, we are observed in the Fabaceae members. Then taxonomy refers to identification, nomenclature, and classification. Identification, classification, and nomenclature. So these three are correct. But here, my dear friends, in this question, two options are there. Definitely identification and nomenclature, correct. And then third one is the classification. Identification, nomenclature, and classification. And assertion based on the formation of the mucilage due to partial dissolution of outer pectin wall in a water. Spirogyra is also called pond silk. Yes, Spirogyra we can call as pond silk. In a matured cell of Spirogyra, the cytoplasm is peripheral with a central vacuole. Yes, both are correct and R is correct explanation to A. And 52 question. Engler and Prontil. Engler is embryophyta and siphonogama. Engler Prontil has classified into plants, embryophyta, siphonogamy. And secondary metabolites used chemosystematics or chemotaxonomy. Then natural system of classification, alpha taxonomy. And nuts belongs to oak tree. And the 53 question, indicate the correct statements from the following. 2, 3, 4 are the correct statement. First one is, 1, 2, 3, 4 is correct statement. 1, 3, 4, 5 correct statement. And the second option is wrong statement. So first one, see here. Cotton is used either alone or in a combination with other fibers in manufacturing all types of the textiles. Yes, right answer. And mercerized cotton is resulted by treating the cotton fibers with caustic soda. Yes. The fuzz, short hairs, is used in preparation of pillows, cushions, and mattresses. Right answer. And cotton treated with nitric acid to produce gunpowder, which is used for making explosives. Yes, it is also right answer. Except second option, remaining all options are correct. Cottonseed oil is used as lubricant, and the oil cake is used as poultry feed. They are not used lubricant, but the oil cake is used poultry. But it is wrong answer. Then the 54th question, the system of classification proposed by the scientist is also called Neo-Adansonian system. Neo-Adansonian system. That is Sokal and Sneath. The system of classification proposed by these scientists is called Neo-Adansonian system. Sokal and Sneath. System of classification. Okay. Then pick up the wrong statement. See here, wrong statement. See here, my dear friends, remember. So this is wrong. Remaining all correct. The rate of production of total organic material is known as gross primary productivity. Yes. The balance energy or biomass remaining after meeting the cost of respiration of producers is called net primary production. Yes, this is also right answer. And the net productivity is equal to gross productivity minus respiration and other losses. Yes, this is also right answer. In 56th question. Several stages of the hydrosphere, along with their suitable examples of given, indicate correct matches. See here, my dear friends. Plankton stage, diatoms. Floating stage, nymphia. Submerged stage, hydrilla. Reed swamp stage, sagittaria. Woodland stage, not juncus, my dear friends. Woodland stage is trees. Woodland stage, that is trees, not juncus. And the addition of consortium or cocktail of microorganisms to the polluted soil is known as, that is bioaugmentation. Bioaugmentation. Addition of consortium of cocktail of microorganism to the polluted soil is known as bioaugmentation. And littoral and swamp forests related to mangroves. Montane wet temperate forests are sholas. Natural grasslands are savannas. And species show a restricted distribution but cover large areas in course of time. That is progressive endemic. Progressive endemic. The study of migration of birds is known as, that is phenology. Very, very important. Phenology. The study of migration of birds is known as phenology. Phenology. Next, assertion. Japanese and Philippines lakes are volcanic lakes in which the extreme chemical conditions are in. Right. A is the right answer. And acid or alkaline lakes associated with the volcanic regions are called volcanic lakes. Yes, this is also correct. So both A and R correct, and R is correct explanation to A. Next, 61. In the fertilized egg of a plant has 40 chromosomes. Remember, fertilized egg has 40 chromosomes. That is zygote. Zygote is condition is 2x condition or diploid condition. So 2n is equal to 40. Remember. And number of chromosomes present in the pollen mother cell. Pollen mother cell. Remember, my dear friends, pollen mother cell is equal to diploid. It is also diploid condition. Then automatically, the fertilized egg contains 40 chromosomes. At the same time, the pollen mother cell is also contain 40 chromosomes. Okay. If the pollen grain is, that is definitely 20 chromosomes. The female gametophyte of typical dicot polygonum at the time of fertilization. Remember, my dear friends, a typical polygonum embryo sac contains definitely female gametophyte or embryo sac contain 8 nucleated 7 celled stage. It is, remember, it is finalized. And 63 question, in cleodic egg, the development of embryo is arrested in freshly laid eggs. And the reason is fleshy laid eggs show drop in temperature, so they need incubation. They need incubation. Both A and R are true, and R is correct explanation to A. Okay. So in cleodic egg, the development of embryo is arrested in fleshy laid eggs, and fleshy laid eggs show drop in temperature, so they need incubation. Right. So both are correct, and R is correct explanation to A. And radial cleavage, we are observed radial cleavage in echinoderms, spiral cleavage observed in annelids, and bilateral cleavages observed in tunicates. Okay, tunicates, and rotational cleavage we are observed mammals. So this is cleavage types and organisms. Embryonic cleavages definitely observed in UGC net, CSIR net, at the same time AP set or TS set also. Then 65th, during different differentiation, different cell types make different steps of proteins at several levels. The following sequence. Number 1, differential gene transcription, differential RNA processing, differential RNA translation, differential protein modification. Okay. And the 66th question, if the deleterious effect of a mutation of a mutation in a gene are overcome by mutation in another gene, the process called as somatic mutation. So somatic mutation, the deleterious effect of a mutation in a gene are overcome by a mutation in another gene. The process is called somatic mutation. Then 67th question. Extra chromosomal inheritance of male sterility was discovered in maize by Rhodes. Rhodes. Okay. That is caused by B chromosomes. Okay. B chromosomes. Extra chromosomal inheritance of male sterility was discovered in maize. Discovered by Rhodes because of this process. B chromosome, that is compared to the shortest chromosome of the normal, compared to normal chromosome. And assertion, mental and motor retardation and death by age 2 to 4 years are phenotypic effect of the individual of Tay-Sachs disease. In the individual of Tay-Sachs disease, galactose 1-phosphate uridyl transferase fails to catalyze transformation of galactose into glucose. Between these two statements, A statement is right answer, but R is false. And adopted characters acquired by an organism are not inherited. They do not get sufficient time to be fixed at the genetic level. So here, A is correct answer, but R is false. Then 17th question. Geological evidence indicates that free oxygen began accumulating in the atmosphere about 2 billion years ago. Yes, this is the right answer. And the free oxygen in the atmosphere is produced by cyanobacteria life forms. Yes, this is also right answer. So between two statements, both are correct, both are true, and R is correct explanation to A. Then 1 question. The records of evolution of birds is sketchy. Then R is the pneumatic bones of birds make poor fossils. So both A and R are correct. R is correct explanation to A. Then 72 question. Animals and their real. Proteopteris, that is Ethiopian, and Opossum, Neo-Arctic, Spinodon, Australian, then prehensile tailed monkey, that is Neotropic. According to Darwinism, which one represents the correct sequence of events in the organ and the origin of new species? That is variation and the inheritance, survival of the fittest, natural selection, struggle for existence. And the second one, survival of the fittest, struggle for existence, natural sectors, natural section, variation and their inheritance. And the third one is natural selection, variation and their inheritance, survival of the fittest, struggle for existence. And the fourth one, struggle for existence, variation and their inheritance, survival of the fittest, natural selection. So this is the Darwinism. Which one is the represent correct sequence? That is, the correct sequence is this one. And the enterokinase stimulates trypsinogen. Enterokinase stimulates trypsinogen. The last one is in the detoxification of ammonia, the key intermediate products, that is the synthesis of citrulline is appeared to be present in the following extrahepatic tissues. That is A and 2, 1 and 2. That is brain and kidney. So this is about my dear friends, remember, this is the AP Set 2012 paper 3 question paper. Remaining question paper will discuss next class. Okay. I wish you good luck.